Bonds are broken and formed during any chemical reaction. A chemical reaction has reactants, which are what goes into the reaction, and these are the things that are altered and changed to make products. Energy has to be added in order to break bonds. It only makes sense that you have to invest energy to break something wide open. Whereas when bonds are formed, energy is released. You can think of it this way. In order to melt ice, you have to invest energy. You have to heat up the ice somehow to get it to melt into a puddle of water. Whereas when you want to freeze water, you have to cool the water down. So in a way, you're removing the heat. Heat's being released by the water as it freezes. Water freezing is exothermic. Exothermic means the reaction releases energy. So the water is releasing heat into the outside environment in order to form ice. Meanwhile, ice melting is endothermic. The reactant, ice, has to absorb heat in order to melt into water, which are the products. If you look at the graph, the products are actually higher up than the reactants, which means that this reaction is endothermic, or it absorbs heat. Activation energy is the energy that needs to be absorbed to start a chemical reaction. It's like pushing over that first domino, which causes the rest of the dominoes to topple over. It's providing that push, or meeting that threshold, which will guarantee that the reaction will start on its own. More often than not, the amount of activation energy required is usually a hurdle to starting the chemical reaction. You either won't have enough of it, or it just takes too long for this to occur. So, in order to help, you can have catalysts, which are usually proteins that speed up a chemical reaction. And it speeds up a chemical reaction by lowering the activation energy. It removes this hurdle. So you can see in the picture there, the frogs on the left have a tough time jumping over that hurdle or that hill. However, catalysts lower the activation energy, and it makes it easier for the frogs to jump over. Once again, just so that we're clear and that we're on the same page, catalysts are usually proteins, or sometimes they're called enzymes, and they lower the activation energy. And you can see there in the graph what a difference it makes. It actually reduces the amount of energy that you have to put in to get the chemical reaction to start going. If you didn't have enzymes or catalysts, the reactions in your body would take millions and millions of years, and you would just get nowhere. You would just end up dying, and you can't carry out important vital functions that you need to survive. Enzymes or catalysts are sensitive to changes in pH and temperature. The optimal temperature for catalysts in your body is usually around 37 degrees Celsius or around 98.6 degrees Fahrenheit. That's the optimal operating temperature for these enzymes. Outside of that temperature, for example, if you get a high fever, this becomes life-threatening because your enzymes start to get denatured. It loses its shape, it starts to unfold, and when it starts to unfold, it no longer has a function. Enzymes work because the active site matches the substrate. So it's kind of like providing an area for the substrate to enter, and if there's a perfect match, the enzyme locks in with it, and then it starts churning out new products. So in the case of an enzyme known as lactase, lactase can break down lactose, which is a complex sugar. So it breaks down lactose into glucose and galactose, which are more simple sugars that the body can actually use for energy. Enzymes require the fit to be perfect. Otherwise, there's no chemical reaction that occurs. This is often compared to the lock and key model, where the substrate acts as the key and the enzyme is the lock. If there's a perfect fit, a chemical reaction takes place and there's products that are made. This concludes my 3-part series on biochemistry. To give you a summary of part 3, just understand that activation energy is the amount of energy required to get a chemical reaction to start. You can lower this activation energy if you have catalysts or enzymes. Catalysts and enzymes are really just special proteins that speed up a chemical reaction. Now, the last thing you have to understand is enzymes have specific shapes. So they have active sites which allow substrates to bind to them. And if there's no perfect fit, then there's no chemical reaction. Thanks for watching, and I'll see you next time on Win Biology.